Okay, good morning, everybody. I hope everybody had a good weekend. Any questions or concerns before I get going with the stuff? Okay, and a reminder for those of you at home, if you got a question, just unmute yourself and pipe up so I can answer you. I think, if I remember right, we left off right with this slide with a comparison of mitosis and meiosis. I felt like it was probably a good idea to maybe go over it one more time just to hopefully solidify it a little bit. So in both instances, meiosis and mitosis, and remember, thinking sex cells and regular body cells, respectively, both of these instances require that the DNA gets replicated before we think about doing any of this separation. All right. Remember, we talked about some of this stuff with the cell cycle already. Do you guys recall where we are doing the DNA replication? What step or stage? Anybody? When we replicate the DNA, when do we synthesize the DNA, make a copy of it? So it happens in part of interphase, and remember, interphase is broken into two gaps, and in between the gap is the S phase. S stands for synthesis. That's when we replicate DNA. When we're pulling in nutrients, when we're just being a normal cell, we got ample stuff, plenty of stuff to replicate the DNA. So all of the specific nitrogenous bases, the sugars, all of that stuff is available. The cell has gone, if we're thinking mitosis, gone through all of the process of ensuring all of its other stuff has been replicated as well. All of the endomembrane system, all of those things get replicated. That technically gets replicated in the first gap when we've got, again, lots of nutrients. Once all of that stuff has happened, one of the checkpoints is basically saying, we're good to go. We've done all of this precursor stuff. We've built extra things, and we've got plenty of ingredients to make DNA. So we synthesize that during interphase, in the S phase of interphase. And then after that, we hit the next gap and then go into either mitosis or meiosis, dependent upon the types of cells, right? And as we described before, there are definitely some steps in here that are similar, and there are a few things that are different. When we think meiosis up at the top, because that's really what we're focusing on at this point, when we think about these guys, they really kind of go through it in two rounds, and we separate it as meiosis 1 and meiosis 2. The first bit of meiosis, the homologous pairs, chromosome number 1 from your father and chromosome number 1 from your mother, both get replicated in the synthesis phase, creating sister chromatids, and those pair up during metaphase 1. And so now you get these homologous pairs. As we bring them together, these homologs have the potential to cross over. That is different than in mitosis. It contributes to variation in the sex cells, and you don't want variation in your body cells. You want those to be copies of one another, clones, if you will. All right. So the first round, we pair up the homologs, and it's random with how they are assorting the 23 pairs. It's not all going to be dad side and mom side. It's mixed as they are kind of shuffled together. All right. So we go through our separation, separating those homologs, and now we've got the sister chromatids still paired together, and that's what gets separated in the next round of meiosis. That is very similar to mitosis, just the separation of those sister chromatids. And in mitosis, we copy all of it. The sister chromatids all line up. We're not homologous pairing, all right? And so they all line up, and we separate the sister chromatids. Now you got two cells that each has 46 chromosomes, if we're talking our own bodies, all right? But up above in meiosis, you wind up with cells that have 23 chromosomes, all right? Haploid, not diploid, all right? And I described as we wrap stuff up Thursday, that ultimately what we're seeing up here with the generation of these 4 cells is really sperm cells where we generate that. And in egg cells, we go through this process, but we ultimately, in the first round of meiosis, one of those cells gets carried on and the other cell gets absorbed by the body, leaving us only with one of these lines. And the same thing happens, and we keep one and the other gets reabsorbed. That keeps the amount of eggs typically to 1, all right, per month. Depends on some circumstances. There are some opportunities for 2 eggs to be released, creating the non-identical twins, all right? But usually it's a single one, all right? So the little table down below compares some of that stuff. That's basically all of the stuff I kind of hit on already, all right? Now we want to think a little bit about problems, all right? Everything we thought about so far is the perfect situation, all right? When things happen as they should. We have talked about issues with mitosis and some problems when we lose some of those checkpoint proteins that are saying you can proceed or you shouldn't proceed. And we start thinking about cancerous situations. There's problems with that when cells divide uncontrolled, unchecked, all right? There's also issues when we think about the replication of these sex cells. There are things that can occur that can lend to problems, all right? And in some circumstances, serious problems, all right? So there are handfuls of things that we really kind of want to think about as we move through here and see some examples, all right? So non-disjunction is a situation, and we'll see this in a second, where we don't separate these chromosomes as we should, all right? And it can be a situation in either the first or the second round of meiosis. So either those homologous pairs in the first round, one of them doesn't separate like it should, so now we pull all 4 to one side. That's a big issue, all right? Or it's in the second round where it's the sister chromatids, and it's the same idea. They get pulled to one side, all right? Either way, you wind up with one cell that's got too much and another cell that is lacking, all right? And then dependent upon which one winds up getting carried on, if it does, that sets the stage for issues down the road, all right? There are other potentials as well. We think about, and we talked about a little more in mitosis, some issues with mutations where you get changes in a base pair. We'll think a little more about that coming up, all right? But a simple mutation is something that also could be occurring in meiosis, something that is heritable, it's passed on to those offspring. That's a potential issue as well. And we could think of it as another little bit up in there as an error in meiosis in the process of synthesizing. We didn't quite replicate as we should, all right? So that's one idea. This is bigger, all right? So inversions and translocations, it's not just like a single base that gets flip-flopped or, you know, an adenine now is a cytosine, something like that, all right? This is big chunks. So a piece of that chromosome with handfuls of nucleotides can potentially get flipped around. That's an issue if we think about reading things in one direction. Now it's all backward, right? And translocations are like crossing over where we see some exchange, but it's not the same chromosome numbers. Translocation is where one chunk of a chromosome winds up on a completely different chromosome. And sometimes it's a fair trade and they swap. Oftentimes it's not like that. And one winds up with a boatload of extra stuff and the other one now is lacking. And it's a similar idea. If we think about instruction manual, now you got stuff that doesn't make any sense. All right. How can I build this stinking shelf? So we showed some karyotypes before the last chapter, I think it was, where we introduced some karyotyping. And this gives us an opportunity to visualize those chromosomes, right? And when we talked about this stuff before, you could see potentials if there's too many or too few, or maybe with some of these translocations, you can see one that's bigger and one that's smaller, all right? There are some opportunities with that. I did mention in a karyotype, you don't see the effect of a single mutation. You don't notice that. You can't tell that there's a cytosine where it should have been an adenine or whatever it might be. Karyotypes don't show you that, right? But they do give you some great insights into the chromosomes and their, how they truly appear. So this is maybe a little more common when we think about staining them up, a couple of different colors, fluorescent tags within there, not something that gives you a whole rainbow of chromosomes. So as we see with these guys, they all sort of look like little X's, right? These are the sister chromatids, right? And the homologous pair, 4 of them together, all right? So when we look at karyotypes, we're looking at these chromosomes after they've already gone through the synthesis phase, all right? That's when we see them. They're paired up like that, and they have this characteristic X-looking appearance, all right? Some bigger and some smaller, but they do look like X's, all right? So this ultimately requires some sorting. They're not paired up like this as we're pulling it out, all right? They're loose, and we have to ensure that we're matching all of these up accurately, all right? And some of the staining that we see in here helps to kind of guide us with that. In this instance, we see 2 X's over here, so we're looking at a female. And as we look at all of these, it's all 2 pairs. There isn't extra or missing chromosomes that we're looking at. So we might say a normal karyotype, right? Again, mutations aren't able to be detected, right? You can see a little bit with some of the staining that you might be able to detect some slight variation, right? Where maybe these bands are a little bit thicker than those bands. And I'm just throwing this out here. I truly don't know, but we might think of a dominant and recessive sort of situation where maybe this gene is not as expressed on the one as it is on the other, all right? Truly don't know if you really could say that based off of some of this, but you might see some variation. And here you can see red, and then red with a little bit of a greenish tip on there, right? A little bit different, right? And you would expect to see some variation between your father chromosome that he donated to you and your mother's chromosome that she donated to you to make you. You would expect that there is some variation within that, all right? Hopefully, all right? Okay. So we can see all sorts of potentials with the karyotype and possibly lending to ideas about questioning what's happening in that unborn child. We could do a karyotype and see, are there genetic anomalies? Are there issues? Are there potential things that we should be concerned about? Maybe we want to be clued into it. Maybe we would like to have the opportunity to make specific decisions. Maybe this child is going to have some severe issues, and we should have the right to be able to make those choices. Is that something that we would like to allow to persist? or is it something that should not? Choices should be ours, all right? Keep that in the back of your mind. Those choices are trying to be taken away. I think that's a big issue, all right? So non-disjunction, again, there's a couple of different potentials, all right? Whether it be the first or the second round, all right? So here we've got them divided into the two different possibilities. The top one happening in meiosis one. So we've got our homologous pairs lined up in metaphase one. And as we go to separate the first ones, the smaller chromosomes, they separate like they should, but these, for one reason or another, don't. And they wind up getting pulled to one side of that cell. Now, as we separate, we wind up with the one that is lacking. That's why we call it N-1, because it's missing one set of chromosome. And over here, it's a +1 because it's got an extra one, all right? And typically, either of these circumstances are going to be bad news, all right? There are a few instances where that cell, that offspring could persist, all right? But oftentimes, if you've got too many or too few, it's the end, all right? Here, we've got the proper stuff happening in meiosis one, separating as they should, but in meiosis two, it doesn't, right? And it's a similar idea. It's +1s and -1s, right? So this one, well, and this one's normal, right? As we move through here, this cell separated, haploid, all fine and good. This cell separated, and we've had a non-disjunction. So one with extra, sister chromatid still there, one missing it, and these ones normal, right? So if we're thinking sex cells in this situation, we've got more issues if it happens in the first round of meiosis, because it impacts all of those. And technically, these down below, the red ones, there's no issues with that. And if it happened to be the egg or the sperm in this instance, there are no problems, right? But if it were either of these, there are issues, right? So it truly depends what step of meiosis or what stage of meiosis the non-disjunction happens in for the impact of these cells, all right? All of them can have problems, or half of them, all right? And again, dependent upon, male or female, there are differences as far as which ones of these would be persisting in the first place, right? So one reason potentially to do a karyotype is to determine if that child potentially has a trisomy. That would be an extra copy of a chromosome, and specifically chromosome number 21. That causes Down syndrome. Let's go back to this. Oops, wrong way. So right here, this chromosome 21, as we look at it, there would be an extra one over here, trisomy, right? So 3 copies, like I said, usually too many or too few aren't going to be viable, but in this circumstance, they are, all right? So Down syndrome is something that's relatively common. It's something that many, probably everybody has seen individuals that have Down syndrome, and there are varying levels of functionality with these people. Some are highly functional, able to do things, work, things like that, kind of live on their own, all fine and good, and others are not, and they're highly dependent, all right? And I'm not a geneticist, but I feel like I might have the answer, all right? When trisomies happen, it can be an entire chromosome, or it can just be a fragment of a chromosome, all right? It doesn't have to be the full piece. If you've got just a little extra chunk of it, it's bad news, all right? And I feel like maybe if you have a small fragment, you might be more functional. And if you've got the entire copy, well, now there's bigger problems, all right? More dependency for sure, right? So having the opportunity to evaluate that gives you a leg up, gives you an opportunity to make some choices and some decisions, all right? This figure shows the risk of Down syndrome and the potential for that risk to increase with age, all right? And it shows the mother's age here. There are some issues with this. It doesn't have to just be the mother, all right? It also doesn't have to truly fit this category or this figure, all right? You could be, young, couple of 20-year-olds, right? And in turn, you could have a child with Down syndrome, all right? Those people, highly unlikely that they would have done any sort of screening, karyotyping. That's not on the radar in the first place, but it still can happen, all right? However, it is considerably more likely as you get older that there are potential issues that can occur. And with the females, this is maybe a little more of a concern because the eggs that they have, not specifically the eggs, but the diploid cells that will in turn create the eggs through meiosis once puberty starts, all of those diploid eggs, germ cells, they're there when you're born. So you have them, they just haven't started this process of meiosis, right? And once puberty starts, that's when this process starts going. So you have a finite amount of cells that are going to go through meiosis that ultimately generates the monthly cycle. With that, as you increase in age, as you get to the point that that basket of eggs is starting to dwindle, you have lived a handful of years and potentially exposed yourself to things that might cause genetic issues, might cause some damage, might lend to some non-disjunction occurring more so than if you're 20, all right? Men constantly are producing these sex cells, all right? It's not like a finite amount that they have. So the potential for issues like that is a little bit minimized, all right? But it's not 100%, all right? And there are definitely instances of old dudes with young women that have children with Down syndrome, all right? And deep down, the finger really ought to be pointing at them, all right? Not at the women, all right? So I kind of have some issues with that because it tends to make it seem as if it's really the woman's issue in the first place. And that's not really 100%, all right? There are definitely, some greater chances with that, but it's the man can have non-disjunction issues as well, and those sperm cells can be likely to contribute it too. So they say at about 35 years old, the incidence of this starts to increase and potentially some screening could be warranted, all right? As we look over here at 40 or 45, which is pushing toward the tail end of your reproductive age, the potential increases pretty dramatically, all right? So maybe you might think about it more along this range, potentially, if you're younger. But like I said, 20-year-olds, you probably really aren't on the radar in the first place to be concerned about it, all right? Maybe not even till you approach 40, all right? And at that point, the doctor might question, is that something you want to do, all right? Do you want to test for it? Deep down in the back of your mind, you need to make those decisions. Is that something that you're able to deal with, or is it something that you would not be able to deal with? And I think realizing that before that sucker ever hatches out is super important, all right? And again, choices, those choices should be yours, right? It should not be dictated to you. Think about that. So It does increase with age and potentially sets the stage for thinking about doing a karyotype, right? That's a sensitive mouse. It bounces all over the place. All right. So we talked about this. Well, we talked about this clear back at the very start of the semester when I was stuck in my basement. I remember that. We talked about the calico cat or the tortoiseshell cat and some differences that we can see. All right. We really should back it up for 1 second and think about, I'm going to back it up to the karyotype, to the chromosomes, all right? All of these chromosomes up here, we've got 2 copies of these. Well, they've replicated, there's 4 of them up here, but in your own cells, we've got 2 copies of all of them, all right? And over here in your sex cells, your X's or your XY, if we're talking male, right? There are some variations. So males get an X and they get a Y, and things seem all fine and good. Females get 2 X's. Things seem all fine and good, right? Females don't technically express both of those X's in every single cell. That's an overexpression, right? You technically only need 1, all right? So what happens? When these cells are first dividing, so upon fertilization, if we are talking female, right, we've got a cell that's got 2 X chromosomes. We start going through handfuls of replications of these cells, creating the little blastula, this handful, 16 or 32 cells. It's a handful of cell replications before it happens. It's not right at the start, all right? So you get a chunk of these cells, all right? You know, a couple dozen or whatever it is. I don't know the exact amount when it happens, all right? But you get these cells, and now, if you are female, 1 of the X chromosomes is going to be turned on, if you will, and the other X chromosome gets taken out of commission, right? There are a bunch of RNA molecules and I think probably some proteins that associate with it, and they basically twist themselves around one of those X chromosomes, taking it out of commission, keeping it condensed, keeping it so that it can't be read, all right? Now you've got a cell that's got the 1 X and the other one shut off, all right? And that's what you want, all right? So in humans, we typically don't really recognize any issues with that stuff, all right? It seems usually all fine and good, all right? Where this cell, this X is on, that cell, the other X is on, whatever it might be, and it's a lot of randomness with that. But it seems to be okay, all right? I will give you an example of that in just a second, or maybe not always, all right? So 1 turned on, 1 turned off. The one that's shut off, we call those bar bodies, all right? A bar body is just this little X chromosome that's been taken out of commission. And you can see these things when you're looking at other, you know, not a karyotype, but you're looking at other genetic stuff. You can see some of those where they've been taken out of commission, all right? So the calico cat is a pretty good example of this, where we can see which X was turned on and which one was turned off, all right? So in these circumstances, we've got a black cat and we've got an orange cat, and love is in the air, right? This mother cat has a handful of kittens, and you might see some orange ones, you might see some black ones, and you probably see some calicos or some tortoiseshells, right? Kind of a mixture, a smattering of both of these colors. And what happens in these cats, one chunk of the coat, the skin, has a specific X chromosome that's activated, the black one. The other patch of skin has another one, the orange one's activated. And you see this patchwork sort of appearance based off of that, all right? This kind of all sorts of black and orange, but definitely differences with these skin cells and the expression. It's whichever X chromosome is turned on, right? The black cat could be the dad, and it gives an X chromosome or it gives a Y chromosome, just depends, right? And in a litter of kittens, there's all sorts of differences with the specific sperm cells, right? Multiple eggs. It's not like a bunch of twins, right? It's there's handfuls of eggs, handfuls of sperm cells that are fertilizing. Each one is an individual as they are born, right? So dependent upon the female cat, right, that gets two X's, the black cat giving the X, the orange cat has her specific X, and then it depends on which of those were the bar body and which weren't. That establishes this patchwork situation, right? Giving us the phenotype, the outward appearance, which oftentimes you can't look at something and say, I know your genotype. I know what your DNA is. Right? It's obvious. Sometimes you can. Calico cat, you can tell, right? This specific pattern, what's happening with that, right? Other examples that might be a little easier would be something like albino. Albinism is a recessive trait, two specific copies, and you can see that phenotypically, all right? So almost always, and we talked about this again at the very start, almost always these cats are females, right? Because in order to get the coloration like this, there has to be two X chromosomes, right? So with non-disjunction, with these issues where chromosomes may fail to separate, there is an opportunity for our little tortoiseshell cat up here to be a male. But in order for that to happen, it's got to have two X's and a Y chromosome. And some of those cells, one X is on, the other cells, another X is on. The Y chromosome's there to make it be male, all right? Almost always in those circumstances, we see very reduced fertility. Sometimes we might say sterility, all right? How do you separate three chromosomes in half? Can't do it very well, all right? With some non-disjunction, there might be some potentials, right? Where you still could get some separation and maybe have a normal cell, all right? So differences, all right? We talked a little bit about that and called that Klinefelter syndrome, two X's and a Y, setting the stage for that, all right? And it is a non-disjunction, all right? There are a couple of others, all right? So this is the same boy with Kreiderschott syndrome at various ages, and this is I think it's a translocation, if I remember right. I don't think it's a extra chromosome. I think it's a jumping gene, if you will, chromosome going to the wrong place. I think the next slide maybe shows that. Maybe I misspoke. So we could see this just a time progression from 2 to 12, right, with that individual. And if I recall, life expectancy is pretty short, right? I don't think much past this with these individuals. Down syndrome that we talked about isn't like that. You can live to a ripe old age in those circumstances, but a lot of genetic anomalies like this cause some big problems. And oftentimes they don't make it out of childhood if they make it in the first place, all right? I want to go back to this real quick while we're talking the X's and the tortoise shell. I told you I was going to give you an example. I technically have a couple of examples that maybe we could share. So there are a few things that we will think about with some genetics. We'll touch maybe a little more in the next chapter with some of this stuff. But when you get your specific genes, we've already mentioned there might be some dominant, there might be some recessive. There are some things that we think about specific disorders that we call X-linked or sex-linked. And those oftentimes have a pretty big impact in males because you only get one copy, right? And in females, to exhibit that, well, you have to have two of those copies. So the mother and the father both contribute this specific gene, right? One example of this is color blindness. And there's a couple of types of color blindness. But an X-linked situation, males are typically more common to be colorblind than females because you only need one copy, right? Females with the two X's, if she has a good copy and a bad copy, pretty much no issues. Carrier and could transmit that bad copy to the offspring, but she's got a good one and a bad one, so it's all fine and good, right? There shouldn't be any issues because there's that good X chromosome, all right? Shouldn't be issues, all right? I had a student many years back. She had a couple of issues that required her to take tests at the ADA office, all right? And there was some interesting things that she shared with me about some of this, and it really was because of top in biology, top in genetics. She wanted to talk a little bit about herself because she was pretty stinking sure she was a genetic oddity, all right? She had some weird stuff going on and was willing to share a little bit. All right, I thought it was pretty interesting. All right, so she had some problems that, again, required her to take tests elsewhere. And one of the things that she needed, and I think I don't know exactly what this was, but she couldn't, when she would just look at a regular page of regular paper, the words would kind of move out and all weird and it's wonky, right? She can't really focus on it because it's almost giving her like three-dimensional stuff. It's just weird, right? But if she put a piece of like blue plastic over the top of it, all of a sudden they all sat down on the page. She could see it. That's pretty crazy, all right? Then we proceed to talk about stuff more than just that, because I'm like, that's crazy, right? And there was I can't remember what she called that, right? The next thing that got me and made me step back a little bit and be like, wait a minute. She was colorblind, two bad copies, all right, to be colorblind. So you would think, all right. She was only colorblind in one eye, not both eyes, right? That's the genetic oddity, right? So that makes me back it up and say, I bet you she is heterozygous for colorblindness because the one eye's got the good X and the other eye had the bad X. That one's expressed colorblind, right? And that gave her the weird depth perception stuff because one's not seeing like it really should be. And as soon as you put a filter on there, all of a sudden it all worked out. Awesome, all right? She had definitely a potential to transmit that color blindness onto offspring if that bad X was concluded, all right? Really kind of odd, all right? So that's one thing we can think about, and an X situation where we may see some impacts. There's other things as well, like hemophilia is another example of a sex-linked situation, more common in males than in females, right? But not unheard of, all right? So I'll tell you one more quick story. I mentioned the Klinefelter with the cat, right? Typically, these circumstances, we don't think of them as being fertile, right? There are opportunities where maybe that male calico could breed with a female calico, and they could pump out a bunch of calicos, and some of those being male, right? But usually, you don't think about it, right? In people, there are some similar ideas with this, and there are a number of different names for some of these sex cell disorders. Too many X's, only one X, a bunch of Y's, or two X's and a Y, right? Whatever it might be. You can have too many sex chromosomes and you can still live, all right? You potentially have some issues, but oftentimes you may not even recognize that you only have a single X chromosome, or you've got two X's and a Y. You may not know it at all right? So unless you did a karyotype, and then you would see it, all right? So here's my story, all right? There was a show, I don't remember what it was, probably like Dateline, something I did not want to watch it, all right? But as I'm scrolling through, I see this and I catch just one little bit that like lured me in, and I'm like, what in the hell is this show, right? So this show, the Dateline, they're following this guy that's telling his story about what has happened to him, all right? How he figured out that he's got Klinefelter's disease, all right? So this guy is a construction worker, and he's out on this job site, and he gets stung by a bee. And he has a little bit of a reaction, whatever, no big deal, all right? Couple of weeks later, he gets stung by another bee and has a severe allergic reaction. Allergies do things like that to you, all right? He's got a severe reaction this time, and he ends up in the hospital. And after that, he recognized some differences starting to happen, all right? And he's figuring it out. Was it the bee sting? Was it something like this? What was it? And I think it was all really, for as far as I can gather, probably random, right? Just because that happened didn't mean it caused this, all right? But this guy, all of a sudden, so it seems, starts to grow breasts. And that's maybe a little bit concerning to this gentleman, all right? And it's something that, maybe he's a little worried about being a construction worker. He's like, oh, all of these guys, they're gonna give me a bunch of crap, right? Whatever it might be, all right? So issues, all right? Wondering what's up here. Why is this happening, all right? The story gets even better, all right? As I start watching this, it's right at about this point where this guy's growing breasts. And I'm like, what is going on? So he proceeds to tell this story where he meets his wife. They are dating. They've been dating for a while. He brings her back to his house, and he decides that he wants to share with her his secret. And he takes her to his room. You'd think love is going to be in the air. I think it was a little bit after. He takes her in and opens his closet and shows her all of his dresses. So this guy, from a very little age, has liked women's clothes. He was a cross-dresser. He thought it was all fine and good. I don't got no problems. Whatever you want to wear, fine, right? So things are progressing with her. He feels like he ought to tell her and show her. He's like, I like to wear ladies' clothes, right? She's okay with that, seems fine. And I joke and I'm like, well, they weren't the same size. That's why he showed her. She's like, I'm not afraid that she's going to steal my dresses. Or maybe he should have found someone. They could have shared clothing, right? I don't know. So doesn't matter. Love is in the air. They get married, right? They also have 2 sons, right? That's something that makes you kind of wonder the littlest bit, all right? So they have 2 sons, then flash forward about a decade, all right? That's when he starts growing breasts, all right? He's got like a 10 and a 7-year-old or something like that. So they've been married a long time. This happens, he's in his probably mid-30s, which with some of the Klinefelter stuff is kind of about when some of these symptoms might start occurring. Really has nothing to do with the bee sting, all right? Maybe a little bit, all right, to stimulate something, but I don't know, all right? So he starts exhibiting these symptoms, growing breasts, wondering what's going on, all right? And goes to the doctor to get some more details. And they're like, well, we could do some genetic screening. Maybe we could get to the heart of the matter, right? So he does this. They do a karyotype. They show him these pictures, 2 X's and a Y. He's got Klinefelter syndrome, right? He is male, right? Y chromosome, right? But he's also growing breasts, which I think made him kind of happy, right? So as soon as he gets this knowledge, it's this aha moment. And he's like, I knew it. That's why I have these tendencies. I am a woman trapped in a man's body. All right. That's what he has decided. He flies to Europe and gets a sex change. All right. Comes back and it shows his wife, her wife, I suppose we should say now, at the airport, and she goes in to give her a kiss and she kind of steps back and I'm like, wait a minute here. What do you do, right? How does this function, right? It ultimately doesn't, right? They got divorced and now these kids have 2 moms, right? Really, their real mom, right, is it. Their other mom, kind of crazy, right? They are torn, right? What do you do? And I think, today, this was, 20 years ago, probably. That's maybe not as unheard of as we think about some of that stuff today. But back then, you're just like, wow, what is happening here? How is it, right? He truly wasn't, just because you have 2 X's and a Y, you are not a female trapped in a male's body, right? But dependent upon your own situations, maybe you are, right? So she ultimately attracted to males. She finds a new partner. The wife finds a new partner. Both live happily ever after. So you would assume, right? I think all fine and good. Crazy, all right? So that brings me back to these boys that they had, all right? He's got 2 X's and a Y. He's got boys, so they both have to have a Y chromosome. They didn't do, at least not in the documentary or whatever, they didn't show any details about these kids, if they did karyotypes or anything like that. But they could be Klinefelter syndrome as well, with 2 X's and a Y, or they could be normal, a single X and a Y. It truly depends. And through non-disjunction, he could have contributed normal sperm, right? Or not, right? And if it's only in those chromosomes, the sex chromosomes, again, you're viable, all right? It's the others that's more of a concern. Really pretty crazy. So these are the inversions and the translocations. So in an inversion, it's the same chromosome, but we flip-flop it, all right? And the DNA is going in a specific orientation, right? Now we flipped it around backward, so it's back, it doesn't make sense. So it's all out of alignment, if you will. On a karyotype, typically aren't going to recognize any of that stuff having happened, right? Just this inversion, because the chromosomes look the same, right? You can't tell that they have been flip-flopped. However, in what we've drawn here, we can see there is a little bit of difference if we can see the specific genetic, the gene segments within there. In the translocation, in this case, this is reciprocal translocation where we are going to exchange between the 2. So this one with that, Matt, so no gain or loss of information still there, but it's not on the right pages, if you will, all right? It's completely different chapters of the book of you, all right? Now they're all mixed up. There's, the information's still there, but trying to access that is going to be a huge issue, all right? That's reciprocal where it's an equal exchange. This one to that one, or, you know, whatever, it's still there. There are others that aren't reciprocal, where a chunk breaks off and gets added to something else, but we don't see it as reciprocal, where, you know, stuff can be lost, right? And that sets the stage definitely for some big problems, right? As we either inversions, translocations, shuttling those. And, sorry, wrong way. When we get back to the specific genetic anomalies, other potential issues where these, you know, autosomal chromosomes, your normal body chromosomes, having too many or too few, the instructions are not there. And it causes ultimately pretty big problems, right? Again, kind of depends on the specifics, right? Any questions or concerns? All right. I am not going to start the next one. I will stop this share. We technically have a few minutes, but I think it's better to just leave you with a clean break with that. And then we will start with, what chapter was that? 7? We'll start with chapter 8 on Thursday. Good? Okay, guys, have a great day. Have a good afternoon.